Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's lesson, I'm going to discuss the different organic compounds which are essential to human life. Organic compounds typically consist of groups of carbon atoms covalently bonded to hydrogen, usually oxygen, and often other elements as well. Created by living things, they are found throughout the world in soils and seas, also in commercial products and every cell of the human body. The four types most important to human structure and function are carbohydrates, lipids, proteins, and nucleotides. Before exploring these compounds, you need to first understand the chemistry of carbon. So let me discuss first the chemistry of carbon. What makes organic compounds ubiquitous is the chemistry of their carbon core. Take note that carbon atoms have 4 electrons in their valence shell, and that the octet rule dictates that atoms tend to react in such a way as to complete their valence shell with 8 electrons. So carbon atoms do not complete their valence shell by donating or accepting 4 electrons. Instead, they readily share electrons via covalent bonds. Commonly, carbon atoms share with other carbon atoms, often forming a long carbon chain referred to as a carbon skeleton. When they do share, however, they do not share all their electrons exclusively with each other. Rather, carbon atoms tend to share electrons with a variety of other elements, one of which is always hydrogen. So carbon and hydrogen groupings are called hydrocarbons. Many combinations are possible to fill carbon's 4 vacancies. Carbon may share electrons with oxygen or nitrogen or other atoms in a particular region of an organic compound. Moreover, the atoms to which carbon atoms bond may also be a part of functional group. So what is a functional group? A functional group is a group of atoms linked by strong covalent bonds and tending to function in chemical reactions as single unit. You can think of functional groups as tightly knit cliques whose members are unlikely to be parted. On the other hand, 5 functional groups are important in human physiology, and these are the hydroxyl, carboxyl, amino, methyl, and phosphate groups. Carbon's affinity for covalent bonding means that many distinct and relatively stable organic molecules nevertheless readily form larger and more complex molecules. Any large molecule is referred to as macromolecule. Macro means large, and organic compounds in this section all fit this description. However, some macromolecules are made up of several copies of single units called monomer. So what is mono? It means one, and mer means part. Like beads in a long necklace, these monomers link by covalent bonds to form long polymers. Poly means many. There are many examples of monomers and polymers among the organic compounds. Monomers form polymers by engaging in dehydration synthesis. This reaction results in the release of a molecule of water, and each monomer contributes, and one gives up a hydrogen atom and the other gives up a hydroxyl group. So polymers are split into monomers by hydrolysis or hydrolysis. So what does it mean by lysis or lysis? It means rupture. The bonds between their monomers are broken via the donation of a molecule of water, which contributes a hydrogen atom to one monomer and a hydroxyl group to the other. This time I would like to introduce to you carbohydrates. The term carbohydrate means hydrated carbon. So what is a carbohydrate? A carbohydrate is a molecule composed of carbon, hydrogen, and oxygen. In most carbohydrates, hydrogen and oxygen are found in the same 2 to 1 relative proportions they have in water. In fact, the chemical formula for a generic molecule of carbohydrates is CH2O. Carbohydrates also are referred to as saccharides, a word that means sugar. 3 forms are important in the body. Monosaccharides are the monomers of carbohydrates. Disaccharides, di meaning 2, are made up of 2 monomers. Polysaccharides are the polymers and can consist of hundreds to thousands of monomers. A monosaccharide is a monomer of carbohydrates. 5 monosaccharides are important in the body. 3 of these are the hexose sugars, so-called because they each contain 6 atoms of carbon, and these are glucose, fructose, and galactose. And the remaining monosaccharides are the 2 pentose sugars, each of which contains 5 atoms of carbon. They are ribose and deoxyribose. The next one is disaccharides. A disaccharide is a pair of monosaccharides. Disaccharides are formed via dehydration synthesis, and the bond linking them is referred to as a glycosidic bond. So what is glyco? It means sugar. 3 disaccharides are important to humans, and these are the sucrose, which are commonly referred to as the table sugar, lactose or milk sugar, and maltose or malt sugars. As you can tell from their common names, you consume this in your diet. However, your body cannot use them directly. Instead, in the digestive tract, they are split into their component monosaccharides via hydrolysis. The next is polysaccharides. Polysaccharides can contain a few to 1,000 or more monosaccharides. 3 are important to the body. The first one is the starch. Starches are polymers of glucose, and they occur in long chains called amylose or branch chains called amylopectin, both of which are stored in plant-based foods and are relatively easy to digest. The second one is the glycogen. It is also a polymer of glucose, but it is stored in the tissue of animals, especially in the muscles and livers. It is not considered as dietary carbohydrate because very little glycogen remains in animal tissues after slaughter. However, the human body stores excess glucose as glycogen, again, in the muscles and livers. And the third one is the cellulose. A cellulose is a polysaccharide that is the primary component of the cell wall of green plants. It is also the component of plant food referred to as the fiber. In humans, cellulose fiber is not digestible. However, dietary fiber has many health benefits. Some of these benefits are it helps you feel full, so you eat less. It also promotes a healthy digestive tract, and a diet high fiber is thought to reduce the risk of heart disease and possibly some forms of cancer. So let me discuss the functions of carbohydrates. The body obtains carbohydrates from the plant-based foods such as grains, fruits, and legumes, and other vegetables provide most of the carbohydrate in the human diet, although lactose is found in dairy products. Most body cells can break down other organic compounds for fuel, and all body cells can use glucose also. Moreover, nerve cells or the neurons in the brain, spinal cord, and through the peripheral nervous system, as well as red blood cells, can use only glucose for fuel. In the breakdown of glucose for energy, molecules of adenosine triphosphate, better known as the ATP, are produced. Adenosine triphosphate or ATP is composed of ribose sugar, adenine base, and 3 phosphate groups. ATP releases free energy when its phosphate bonds are broken and thus supply ready energy to the cell. More ATP is also produced in the presence of oxygen than in pathways that do not use oxygen. The overall reaction for the conversion of energy in glucose to energy stored in ATP can be written as C6H12O6 plus 6O2 arrow 6CO2 plus 6H2O plus ATP. In addition to being a critical fuel source, carbohydrates are present in very small amounts in cells' structure. For instance, some carbohydrate molecules bind with protein to produce glycoproteins and others combine with lipids to produce glycolipids, both of which are found in the membrane that encloses the contents of the body cells. So let's move on to the next, which are the lipids. So what is a lipid? A lipid is one of a highly diverse groups of compounds made up mostly of hydrocarbons. The few oxygen atoms they contain are often at the periphery of the molecule. Their nonpolar hydrocarbons make all lipids hydrophobic. And what does it mean? In water, lipids do not form a true solution, but they may form an emulsion, which is the term for a mixture of solutions that do not mix well. The next one are the triglycerides. So what is a triglyceride? A triglyceride is one of the most common dietary lipid groups and the type found most abundantly in body tissues. This compound, which is mostly referred to as a fat, is formed from the synthesis of two types of molecules. A glycerol backbone at the core of the triglycerides consists of 3 carbon atoms, 3 fatty acids, long chains of hydrocarbons with a carboxyl group and methyl group at the opposite ends extend from each of the carbon of the glycerol. In this picture are the triglycerides. Triglycerides are composed of glycerol attached to the 3 fatty acids via dehydration synthesis. Take note that glycerol gives up a hydrogen atom and the carboxyl groups on the fatty acids each give up a hydroxyl group. Triglycerides formed via dehydration synthesis. Glycerol gives up hydrogen atoms from its hydroxyl groups at end bond, and the carboxyl group on each fatty acids chain gives up a hydroxyl group. A total of 3 water molecules are thereby released. In this picture is the fatty acid shapes. The level of saturation of fatty acid affects its shape. In figure A, saturated fatty acids chain are straight, and in the figure B, unsaturated fatty acids chain are kink. So fatty acid chains that have no double carbon bonds anywhere along their length and therefore contain the maximum number of hydrogen atoms are called saturated fatty acids. These straight rigid chains packed tightly together are solid or semi-solid at room temperature. Butter and lard are examples, as the fat found on a steak or in your own body. In contrast, fatty acids with 1 double carbon bond are kink at that bond. These non-saturated fatty acids are therefore unable to pack together tightly and are liquid at room temperature. Polyunsaturated fatty acids contain 2 or more double carbon bonds and are also liquid at room temperature. Plants' oils, such as olive oil, typically contain both mono and polyunsaturated fatty acids. Whereas a diet high in saturated fatty acids increases the risk of heart disease, a diet high in unsaturated fatty acids is thought to reduce the risk. This is especially true for the omega-3 unsaturated fatty acids found in cold water fish such as salmon. These fatty acids have their first double carbon bond at the third hydrocarbon from the methyl group, which is referred to as the omega end of the molecule. Finally, trans fatty acids found in some processed foods, including some stick and tub margarines, are thought to be even more harmful to the heart and blood vessels than saturated fatty acids. Trans fats are created from unsaturated fatty acids such as corn oil when chemically treated to produce partially hydrogenated fats. As a group, triglycerides are a major fuel source for the body. When you are resting or asleep, a majority of the energy used to keep you alive is derived from the triglycerides stored in your fat or adipose tissues. Triglycerides also fuel long, slow physical activities such as gardening or hiking and contribute a modest percentage of energy for vigorous physical activity. Dietary fat also assists the absorption and transport of the nonpolar fat-soluble vitamins such as vitamin A, D, and E, and also vitamin K. Additionally, stored body fat protects and cushions the body, bones, and internal organs and acts as insulation to retain body heat. Fatty acids are also components of glycolipids, which are sugar fat compounds found in the cell membrane. Lipoproteins are compounds in which the hydrophobic triglycerides are packaged in protein envelopes for transport in body fluids. This time, let's move on to phospholipids. As its name suggests, a phospholipid is a bond between the glycerol component of a lipid and phosphorus molecule. In fact, phospholipids are similar in structure to triglycerides. However, instead of having three fatty acids, a phospholipid is generated from a tri or diglyceride, a glycerol with just two fatty acid chains. The third binding site on the glycerol is taken up by the phosphate group, which in turn is attached to a polar head region of the molecule. Take note that the triglycerides are nonpolar and hydrophobic. This still holds for fatty acid portion of a phospholipids compound. However, the phosphate containing group at the head of the compound is polar and thereby hydrophilic. In other words, one end of the molecule can interact with oil and the other end with water. This makes phospholipids ideal emulsifiers, compounds that help disperse fats in aqueous liquids and enables them to interact with both the watery interior of cells and the watery solution outside the cells as components of the cell membrane. In the figure A of this picture, phospholipids are composed of two fatty acids, glycerol and phosphate group. In the figure B, esterols are ring-shaped lipids. Shown here is cholesterol. And in figure C, prostaglandins are derived from unsaturated fatty acids. Prostaglandin E2 includes hydroxyl and carboxyl groups. Next is we have steroids. So what is a steroid? A steroid is compound referred to as asterol. Has its foundation a set of 4 hydrocarbon rings bonded to a variety of other atoms and molecules. Although both plants and animals synthesize esterol, the type that makes the most important contribution to human structure and function is cholesterol, which is synthesized by the liver in human and animals and is also present in most animal-based foods. Like other lipids, cholesterol's hydrocarbons make it hydrophobic. However, it has a polar hydroxyl head that is hydrophilic. Cholesterol is an important component the bile acids, compound that help emulsify dietary fats. In fact, the word root chole refers to bile. Cholesterol is also a building block of many hormones, signaling molecules that the body releases to regulate processes as or at distant sites. Finally, like phospholipids, cholesterol molecules are found in the cell membrane where their hydrophobic and hydrophilic regions help regulate the flow of substances into and out of the cell. The next is prostaglandins. Like a hormone, a prostaglandin is one of a group of signaling molecules, but prostaglandins are derived from unsaturated fatty acids. One reason that the omega-3 fatty acids found in fish are beneficial is that they stimulate the production of certain prostaglandins or prostaglandins that help regulate aspects of blood pressure and inflammation and thereby reduce the risk for heart disease. Prostaglandins also sensitize nerves to pain. One class of pain-relieving medication called non-steroidal anti-inflammatory drugs works by reducing the effects of prostaglandin. This time, let's move on to proteins. You might associate proteins with muscle tissue, but in fact, proteins are critical components of all tissues and organs. So, what is a protein? A protein is an organic molecule composed of amino acids linked by peptide bonds. Proteins include the keratin in the epidermis of the skin that protects underlying tissues, the collagen found in the dermis of the skin, in bones, and in meninges that cover the brain and the spinal cord. Proteins are also components of many of the body's functional chemicals, including digestive enzymes in the digestive tract, antibodies, the neurotransmitters that neurons use to communicate with other cells, and the peptide-based hormones that regulate certain body functions, for instance, growth hormone. While carbohydrates and lipids are composed of hydrocarbons and oxygen, all proteins also contain nitrogen and many contain sulfur in addition to carbon, hydrogen, and oxygen. So this is the microstructure of proteins. Proteins are polymers made up of nitrogen-containing monomers called amino acids. So what is an amino acid? Amino acid is a molecule composed of an amino group and carboxyl group together with a variable side chain. Just 20 different amino acids contribute to nearly all of the thousands of different proteins important in human structure and function. Body proteins contain a unique combination of a few dozen to a few hundreds of these 20 amino acid monomers. All 20 of these amino acids share similar structure, and all consist of a central carbon atom to which the following are bonded. Number 1 is a hydrogen atom. The next one, or number 2, is an alkaline amino group. And number 3, an acidic carboxyl group. And the last one, or number 4, is a variable group. Take note that all amino acids contain both an acid and the base, which is amine or nitrogen containing. For this reason, they make excellent buffers, helping the body regulate acid-base balance. What distinguishes the 20 amino acids from one another is their variable group, which is referred to as a side chain or an R group. This group can vary in size and can be polar or nonpolar, giving each amino acid its unique characteristics. For example, the side chains of two amino acids, cysteine and methionine, contain sulfur. Sulfur does not readily participate in hydrogen bonds, whereas all other amino acids do. This variation influences the way that proteins containing cysteine and methionine are assembled. In this picture is a peptide bond. Different amino acids join together to form peptides, polypeptides, or proteins via dehydration synthesis. The bonds between the amino acids are peptide bonds. Amino acids join via dehydration synthesis to form protein polymers. The unique bond holding amino acids together is called peptide bond. So what is a peptide bond? A peptide bond is a covalent bond between two amino acids that forms by dehydration synthesis. A peptide, in fact, is a very short chain of amino acids. Strands containing fewer than 100 amino acids are generally referred to as polypeptides rather than proteins. The body is able to synthesize most of the amino acids from components of other molecules. However, 9 cannot be synthesized and have to be consumed in the diet. These are known as the essential amino acids. 3 amino acids available for protein construction are said to reside in the amino acid pool within cells. Structures within cells use these amino acids when assembling proteins. If a particular essential amino acid is not available in sufficient quantities in amino acid pool, however, synthesis of protein containing it can slow or even cease. This picture here is the shape of protein. In figure A, the primary structure is the sequence of amino acids that make up the polypeptide chain. And in the figure B, the secondary structure, which can take the form of an alpha helix or a beta-pleated sheet, is maintained by hydrogen bonds between amino acids in different regions of the original polypeptide strand. And in figure C, the tertiary structure occurs as a result of further folding and bonding of the secondary structure. And in figure D, the quatermany structure occurs as a result of interaction between 2 or more tertiary subunits. The example shown here is hemoglobin, which is a protein in red blood cells which transports oxygen in the body tissues. Just as a fork cannot be used to eat soup and a spoon cannot be used to spare meat, a protein shape is essential to its function. A protein shape is determined most fundamentally by the sequence of amino acids of which it is made. The sequence is called as the primary structure of the protein. Although some polypeptides exist as linear chains, most are twisted or folded into more complex secondary structure that form when bonding occurs between amino acids with different properties at different regions of the polypeptide. The most common secondary structure is a spiral called an alpha helix. If you were a length of string and simply twist it into a spiral, it would not hold the shape. Similarly, a strand of amino acids could not maintain a stable spiral shape without the help of hydrogen bonds, which create bridges between different regions of the same strand. Less commonly, a polypeptide chain can form a beta-pleated sheet in which hydrogen bonds form bridges between different regions of single polypeptide that has folded back upon itself, or between 2 or more adjacent polypeptide chains. The secondary structure of proteins further folds into a compact three-dimensional shape, referred to as the protein's tertiary structure. In this configuration, amino acids that had been very distant in the primary chain can be brought quite close via hydrogen bonds, or in proteins containing cysteine via disulfide bonds. So what is a disulfide bonds? A disulfide bond is a covalent bond between sulfur atoms in a polypeptide. Often, 2 or more separate polypeptides bond to form an even larger protein with a quatermany structure. The polypeptide subunits forming a quaternary structure are different. For instance, hemoglobin, the protein found in red blood cells, is composed of 4 tertiary polypeptides, 2 of which are called alpha chains and 2 of which are called beta chains. When they are exposed to extreme heat, acids, bases, and certain other substances, protein will denature. Denaturation is a change in the structure of molecule through physical or chemical means. Denatured proteins lose their functional shape and are no longer able to carry out their jobs. An everyday example of protein denaturation is the curding of milk when acidic lemon juice is added. So the contribution of the shape of a protein to its function can hardly be exaggerated. For example, the long, slender shape of protein strands can make up muscle tissue is essential to their ability to contract and relax. As another example, bones contain long threads of protein called collagen that acts as scaffolding upon bone minerals are deposited. These elongated proteins called fibrous proteins are strong and durable and typically hydrophobic. In contrast, globular proteins are globes or spheres that tend to be highly reactive and are hydrophilic. The hemoglobin proteins packed into red blood cells are an example of this. However, globular proteins are abundant throughout the body, playing critical roles in most body functions. Enzymes introduced earlier as protein catalysts are examples of this. So this ends the part 1 of this topic, which is the organic compounds that are essential to human life. If you have any question, please write it in the comment section down below. And once again, I would like to thank you all for watching this video and for listening to my lecture. And until my next video, bye everyone.